Okay, chapter 7 deals with microbial metabolism, and these are simply the chemical reactions that support the life processes critical to allowing these microbes to reproduce and grow and do their job in the environment. And they're made up of two broad sets of reactions, anabolic and catabolic reactions. Anabolic reactions build large molecules from smaller pieces and consume energy in that process, usually in the form of ATP, while catabolic reactions tear molecules apart and release energy, which is then captured and transferred to ATP so that it can be used to do work. Okay, so we're going to talk about the biochemical processes critical for life, how bacterial metabolism interacts with our health, and how microbial photosynthesis and nitrogen fixation support life and the role it has in the ecosystem and how this impacts our health as well. In this section, we'll talk about um, basically metabolism and some of the variations that microbes can use in order to carry out metabolism. We'll explain the importance of energy and entropy in living cells, and we'll state the role of enzymes in controlling biochemistry. Okay, so the ultimate source of life energy for the planet comes from the sun. The the sun produces electromagnetic radiation, which covers a wide range of wavelengths. And for photosynthetic organisms, they can take within the visible portion of the UV spectrum the wavelengths for red and blue light, and then using the energy from that, cause CO2 and water to combine in an organelle called the chloroplast in eukaryotic organisms, okay, and also in the cell proper in photosynthetic prokaryotes, okay, and form carbohydrates. And then those carbohydrates can in turn be torn apart in order to generate energy for the organism to live, okay. Um, a lot of the energy that hits the earth in the form of solar energy is lost as heat. Okay, the rest is used for growth and work and reproduction. Okay, and then because the sun is continuously bombarding the earth with this energy, life can continue. Right? If the sun were to go out, we would all die for several reasons, not the least of which is the end of photosynthesis on the planet, okay, which means it would be ultimately in the ecosystem no source of food for organisms that are um, dependent on energy containing organic molecules to survive, right? And that would likely cause the collapse of life on the planet to a large extent. Not to mention the fact that the temperature shift would likely kill everything. Okay, so the sun is our friend, and it is the master switch in determining the climate on our planet as well. Okay, if the sun's energy were to decrease or increase by just 10%, um, life on planet Earth would be dramatically changed. Okay, we could go from a global ice ball to a global desert with just those tiny amounts of change in the amount of solar energy. And it is, in fact, the tilt of the Earth on its axis and the way that we move around the sun that develops the seasons in the northern and the southern hemispheres, right? Just that tiny change in distance from the sun and in tilt away from or towards the sun has a dramatic effect on the weather, okay? So you can see how important the sun is. Okay. Um, Photosynthesis is the key. Photosynthetic organisms, whether they be prokaryotic or multicellular, such as in plants, they are responsible for turning that energy into a form that can be used by most of the rest of the ecosystem. The microbial and plant biomass enters heterotrophs and decomposers, and they convert a small fraction of that biomass at each successive level, and the rest of the energy gets lost as heat and as work. Okay, so remember that we can have a variety of different setups in single-celled organisms in order to carry out what we think of as metabolism. Okay, phototrophs use light and high-energy molecules to donate electrons as acceptors. Examples would be photosynthetic organisms. Chemotrophs use high-energy food molecules that donate electrons to an acceptor. In organotrophy, for organisms that are aerobic, they need high-energy organic molecules and oxygen to do their job, and the equation basically looks like the reverse of photosynthesis, but keep in mind that those chemical reactions are carried out by different enzymes and usually in different locations in the cell. Okay, organotrophic anaerobic organisms are going to utilize high energy electrons from organic molecules, but the terminal electron acceptor is not going to be oxygen. Okay, oxygen in many cases is toxic to these organisms. Okay, so the organic molecule donates electrons to itself or to some other molecule. An example here is fermentation, in which sugars are broken down and we generate carbon dioxide and then ethanol, okay, which is basically a semi-oxidized form of an organic molecule called ethane, or you could look at it as a reduced form of acetic acid. Okay. In lithotrophy, inorganic molecules donate electrons to oxygen. Okay, so those are going to be reduced metals that are going to become metal oxides, or they're going to be reduced inorganic compounds that are going to become oxidized. Okay, and the oxygen will be converted into water as it takes on 2 protons and a couple of electrons. And then in lithotrophic anaerobic organisms, the electrons are again donated by an inorganic compound, and then they are transferred to something other than oxygen. Okay, in this case, in methanogenesis, they're donated to the carbon and carbon dioxide to make methane. Okay, so the bottom line here is that in every equation, what we're looking at on the left-hand side of the equation is a higher energy content than what we're looking at on the right side of the equation. So where does the extra energy go? A lot of it gets lost as heat. Some of it gets lost as work and movement, for instance. And so at the end of the day, this still ends up being an extremely efficient process compared to something like a man-made engine. Okay. In order to make complex cells, organisms have to utilize energy, right? And so they have to take in something that has energy content, right? Move that energy to something that can be used for work, right? And then as the work gets done, we build the organism, we repair the organism, we grow the organism, and we give off heat. Okay. And we do work at the same time. Okay, so in biology, the enzymes that carry these chemical reactions out are proteins whose job it is to speed up chemical reactions inside the cell so that they give us the products that the cell needs quickly enough for them to be useful. Okay, the nice thing about an enzyme is that it isn't destroyed in the chemical reaction, and so it can be reused over and over again. Okay, and this way the chemical reaction speeds up without us having to worry about changing the temperature or changing the concentration of the reactants, which are the things that are going to form the product. Okay, because if we did that, we might create conditions that would kill the cell. Okay, we've talked about anabolism and catabolism. Okay, and the role of the enzymes. Basically, the reason they speed up the chemical reaction is through the lowering of something called activation energy, which is basically the energy needed to produce the collision or the wrenching apart that makes and breaks chemical bonds so that the reaction can proceed. Okay, and so it makes it easier to go from a reactant to a product, which means that we speed up the chemical reaction significantly. These chemical reactions, those that are what we would call exergonic, the ones that give off energy when they proceed, would happen on their own, okay, but they would be much slower because we would be waiting on things like random collisions in order for those reactions to take place, okay? For chemical reactions that are endergonic, meaning energy consuming, we need energy from some other reaction, usually an exergonic reaction, in order for the reaction to proceed, okay? And so enzymes let that happen, right? Enzymes can steal energy from an energy yielding reaction and then use that energy to do the work of an energy consuming reaction, okay? And generally that energy is provided in the form of ATP, okay? So we'll talk about catabolism and catabolites, and we'll talk about high energy carriers like ATP and NADH, and energy yielding and energy spending reactions, okay? So we talked earlier about catabolism and anabolism, right? In catabolism, we take molecules and we tear them up, and the energy that comes out from splitting the chemical bonds is going to be captured and transferred to a useful molecule that can be used to do work right away, and that molecule is generally ATP, okay? In anabolic reactions, we take energy, usually from ATP, and use that to make something, okay? We might make a complex carbohydrate, or we might make a protein, or we might make nucleic acids, just to name a few molecules, okay? So the catabolic reactions power the anabolic reactions. In oxidation reduction reactions, which we talked about in the chemistry chapter, um, the, the, the element or the molecule that's oxidized is going to lose potential energy, while the element or molecule that's reduced is going to gain potential energy. And the potential energy exists in the electrons of the chemical bonds, okay? Or in the case of an element, it exists in the outer shell electrons, all right? So reduced means high energy, oxidized means less energy, okay? Biomolecules can donate electrons and become oxidized when they do that, all right? Electron carriers like NAD, FAD, and ATP pick up electrons from another source and as a result pick up energy, okay? Oxidation, donating the electrons, so losing electrons to something else. Reduction, gaining electrons from another source, okay? One molecule of NADH carries about 3 times as much energy as a molecule of ATP. One molecule of FADH2 carries about twice as much energy as a molecule of ATP, okay? So those are energy equivalencies, all right? So what you're looking at here is just an example of some of the energy transfer reactions that take place, okay? In A, the chemical reaction of ADP with inorganic phosphate makes ATP. This reaction requires energy input because negatively charged oxygens on the phosphate are forced to be near each other. Removal of each phosphate by hydrolysis using water is going to let the energy pop out, okay? In the reduction of NAD, the ring in pink loses a double bond as 2 electrons are gained from an electron donor. The resulting NADH is going to carry energy. The reaction is going to consume 2 hydrogen atoms One bonds to NAD, forming NADH, while the other ionizes in solution, forming a proton. Okay, so an easy way to do this, all right, is if you're looking at a molecule, an organic molecule, okay, and you want to know which one has the most chemical potential energy, you just count the number of carbon-hydrogen bonds, okay? So in the case of NAD, for instance, if you count the number of carbon-hydrogen bonds on the group that does the work here, this little group here, which is attached to the rest of a dinucleotide, okay, you see that there's, and again, the hydrogens aren't drawn in here, they're implied. There's one hydrogen here, one here for 2, one here for 3, and one here for 4, okay? And then if we go over to the reduced version, what do we have? We have 1, 2, 3, 4, 5, okay? So this has more chemical potential energy than this does, okay? So anytime you see an organic molecule pick up double bonds, okay, or pick up oxygen, all right, then you're going to say that it's been oxidized, okay? In the case of ATP, the power is in a different place, okay? The power is in these phosphate bonds. This is the money part of the molecule here, and as you crack these bonds, considerable energy gets released, okay? You can see an example here of transformation of matter in catabolic reactions, okay? All reactions are going to be catalyzed by enzymes that work on specific substrates. An example here are polysaccharides broken down into disaccharides and monosaccharides. An example being glucose. Glucose and other sugars are converted into pyruvate, which releases acetyl groups and acetate, and the breakdown of these products of fatty acids, amino acids, and other plant materials such as lignin, and it can also yield the energy, okay? So again, look at what's happening over here, right? You've got this big long molecule, many monosaccharides hooked together in a big old chain, right? And then what we do is we crack them, right? We crack the chain. When we do that, energy comes out just in the cracking of the chain. And then we can crack these little bits of chain again, more energy comes out, and then we can take these monosaccharides and then we can crack the ring, okay, and generate something called pyruvate, and we strip energy in that process and give it to NADH and ATP in a process known as glycolysis, and we'll get the details on that in a bit. And then we generate a molecule called pyruvate, which enters the Krebs cycle in many organisms, and then we can crack the chemical bonds in the pyruvate between the carbons and the hydrogens, spit out CO2 as a low energy end product, and then transfer the energy that's in the carbon hydrogen bonds in the pyruvate to something called NADH or FADH2, right? And then we can oxidize those molecules in something called the electron transport chain, okay? And then there are other alternate pathways, right? You can break down proteins into amino acids, and you can convert those into what are called keto acids by stripping out the nitrogen and replacing it with a keto group, okay? Or you can do the reverse. If you're doing a buildup reaction, you can take keto acids and turn them into amino acids and use those to build proteins, okay? And that's done by a different set of enzymes, usually in a different area of the cell, okay? So these are breakdown pathways, all right? And going in the reverse would be an anabolic pathway, a buildup pathway, right? You see the same thing here with lipids, right? You can generate glycerol and fatty acids. The fatty acids can feed in to the Krebs cycle as units of acetyl-CoA, and the glycerol can be converted into pyruvate, and that can enter the Krebs cycle as well, okay? So all sorts of ways that we can basically pull the energy out of the carbon-hydrogen bonds, the carbon hydroxide bonds, okay, and transfer it ultimately to ATP, okay? So we'll talk about glycolysis and the Krebs cycle, and then we'll talk about how we can use chemical tests to ID pathogens. Okay, so in the big three legs of what we call oxidative metabolism, um, we have enough energy to let the cell grow and reproduce and repair itself and do its job effectively in the environment. Okay, um, glycolysis, the Krebs cycle, and the electron transport chain together generate approximately, okay, 38 ATPs. Okay, that number can vary a bit based on genetics. Okay, um, but that's the approximate number. In the absence of oxygen, that same set of pathways will generate a whopping 2 ATPs in processes like fermentation or lactic acid metabolism. Okay, so the presence of oxygen definitely gives you more bang for your buck in an organism that can carry out either type of pathway. Okay, so those would be things like facultative aerobes or anaerobes. All right. In the first of the three legs, in glycolysis, a 6-carbon molecule is split into two 3-carbon molecules, which are then fed into the carboxylic acid cycle. Okay, the molecule that feeds in is glucose. The end product is something called pyruvate. All right, and on the way from glucose to pyruvate, we kick out some NADH, which will later be oxidized to make some ATP in the electron transport chain, and we generate some ATP directly. Okay, so it's a fast pathway, but it doesn't yield a lot of energy. Okay, on its own. A lot of bacteria use a glycolytic pathway called the Embden-Meyerhof pathway. Glycolysis happens in two phases, an energy consuming and an energy releasing phase. So let's take a look at glycolysis, and later we will talk a little bit about the Krebs cycle.
Glucose can be completely broken down to yield energy through a few consecutive metabolic pathways, beginning with glycolysis. In glycolysis, glucose, which has 6 carbons, is split into 2 molecules of pyruvate, which have 3 carbons each. Overall, a small amount of energy is released, some of which is captured by energy-carrying molecules, NADH and ATP. Here the reactions of glycolysis will be arranged vertically based on the energy content of the substrate molecules. Glucose contains more energy than the 2 pyruvate molecules combined, which is why energy-carrying molecules can be generated during glycolysis. Glycolysis occurs in 2 stages. Stage 1 actually consumes energy, 2 ATP molecules, in order to prepare glucose for a controlled breakdown that ultimately releases more energy than is invested here. In stage 1, 2 hydroxyl groups on glucose are replaced with phosphoryl groups from 2 ATP molecules, and the molecule is slightly rearranged. The resulting molecule is called fructose 1,6-bisphosphate. This sugar then splits into 2 3-carbon molecules of glyceraldehyde 3-phosphate, each with a single phosphoryl group. These substrate molecules are primed to start releasing energy. In stage 2 of glycolysis, each of the 2 G3P molecules loses 2 electrons from 2 hydrogen atoms, converting NAD+ to NADH. NADH is a reducing energy carrier. The oxidation of G3P releases enough energy to add inorganic phosphate to each molecule, forming 2 molecules of 1,3-bisphosphoglycerate. Subsequent reactions transfer both phosphoryl groups from each molecule to a total of 4 ADP molecules, making 4 ATP molecules. Notice that although 2 ATP molecules were consumed in stage 1, 4 ATP molecules are produced in stage 2, for a net generation of 2 ATP molecules from glycolysis. At the end of glycolysis, the remaining 3-carbon molecule, pyruvate, no longer carries phosphoryl groups, but still contains a significant amount of energy that can be harvested by further oxidation. Under
Okay, So the Embden-Meyerhof pathway of glycolysis and the Enter-Deuteroff pathway, um, also known as the pentose phosphate shunt, um, are some alternate ways of stripping energy out of carbohydrates and transferring them to either NADH or NADPH or directly to ATP. All right. So in the Embden-Meyerhof pathway, all right, we go from glucose to glucose 6-phosphate to fructose 6-phosphate to fructose 1,6-bis, and then we go phosphoglyceraldehyde to dihydroxyacetone phosphate, then phosphoenol pyruvate, and then finally we end up at pyruvate. Okay, and the pyruvate is going to enter the electron transport chain, being converted to something called acetyl-CoA, and that is an energy yielding process as well. And then the acetyl-CoA will become oxidized in one turn of the Krebs cycle. All right. So with glucose, what happens is that you run this pathway twice, right? And both of those pyruvates end up going into the Krebs cycle. Now, in the Enter-Deuteroff pathway, we have sugars that can be converted into 6-phosphogluconate, all right? And then they can be converted into 2-oxy, 3-deoxy, 6-phosphogluconate, which can be converted into glyceraldehyde 3-phosphate. And from that point on, we can go through glycolysis and dump into the Krebs cycle, right? Certain organic acids can be converted to 2-oxo, 3-deoxy, 6-phosphogluconate and enter glycolysis at the same point, okay? And then in the pentose phosphate shunt, what happens is that ribulose 5-phosphate, okay, can end up being converted into sugar phosphates that have a variety of different carbons associated with them, and some of those can enter the glycolytic pathway at the level of glyceraldehyde 3-phosphate as the 3-carbon species. Okay. Another thing that can happen with these sugar phosphates is they can be employed in biosynthesis, all right, and that can build some of the structures of the organism that are composed of things like carbohydrates, or if we bring some nitrogen into the equation, we can make some amino acids and build some proteins, okay? So again, different enzymes are doing these jobs, okay? Ribulose 5-phosphate and NADPH can be basically siphoned off from the Enter-Deuteroff pathway, and generating these sugar phosphates allows us to throw some energy at glycolysis and then use some of it for biosynthesis, okay? And then there's going to be some ATP and NADPH, which is similar to NADH, generated along the way as well. Okay? In fermentation, electrons are removed from the donor molecule and reduce NAD to NADH, producing 2-ATP. The NADH is going to send the electrons to the electron transport chain, okay? And that would be in respiration, but in fermentation, the NADH turns around and gives the electrons to pyruvate and can generate either lactate or ethanol, okay? And those can eventually be oxidized later to generate energy in the event that we end up in a high oxygen situation, or if we're strictly on the fermentation side of metabolism, we don't do any electron transport chain work, then that is the end of our energy producing pathway, right? And the result then is that the anaerobic or fermentative organisms that only use that pathway for growth grow very slowly, okay, because of the low energy yield that accompanies it. And then the byproducts can be useful, right? The alcohol can be used to generate beverages. We use fermentation from yeast, which is a eukaryote, to do things like make beer and bread. Okay, the beer utilizes the alcohol that comes from the fermentation. The bread um, generates alcohol, but also CO2, and it's the CO2 that makes the bread rise, and the alcohol gets baked out of the bread during the course of the baking process. Right, it just vaporizes. In respiration, what happens is we go through glycolysis and generate pyruvate, and NADH and ATP are produced along the way, right? And then the pyruvate enters the electron transport chain. No, sorry, the pyruvate enters the citric acid cycle as acetyl-CoA and gets oxidized in a stepwise fashion, producing some ATP directly, but also FADH2 and NADH, which will also eventually be oxidized by the electron transport chain. And it's the electron transport chain that is going to produce the bulk of the ATP in these three legs of metabolism. It's estimated that it produces about 32 of the 38 ATP that we get from adding all of the ATP production of metabolism together: glycolysis, the citric acid cycle, and the electron transport chain. Okay, you can see some fermentation pathways that happen in bacteria and yeast. Okay, the purpose of shifting into fermentation in a bacteria or a yeast that can do either aerobic or anaerobic metabolism is to generate enough NAD in order to keep glycolysis going. Okay, and then those byproducts, be it alcohol or lactate, okay, can end up being utilized later, or they can be siphoned off as products for use in things like making beer or bread or yogurt. Okay, you can see in the fermentation pathways some of the products that can result. Right, formic acid is an example. Right, ethanol is another one. Lactate, right, another example. Succinate. Okay, and then if we stay in the oxidative pathway and go straight down, we can also have alternative pathways off of acetyl-CoA, right, which is produced as we attempt to enter the electron transport chain, but we can generate from that things like acetoacetic acid, which can be converted to acetone and isopropanol, right, in some organisms, or butyryl-CoA, which can end up forming butanol, all right? And again, the purpose of all of these is to produce some oxidized electron carriers so that we can continue to run glycolysis. Okay, that's the key. You need NAD in the early stages of glycolysis for glycolysis to happen, right? And if we don't have any NAD, then we can't run the pathway. So all of these guys end up, as you can see, spitting out NAD in order to keep the pathway going. And the byproducts may or may not be useful to the organism. Okay. Alternative pathways. Examples include phosphoenol pyruvate, sometimes abbreviated as PEP, to send products, many of which we use in food or industry. Different species do different portions of the pathway. So again, not every organism can pull off all of these different tricks. Okay, all depends on the enzymes that you have. You can detect fermentation end products using certain types of media. Okay, in panel A, right, you can see organisms cultured in a tube of broth containing phenol red. This is panel A over here on the left. Okay, the dye turns yellow when protonated at low pH, so when it gets acidic it turns yellow. Phenol red is orange and red at neutral pH, and yellow at an acidic pH, and red at basic pH. A small inverted tube collects gas products, such as carbon dioxide and hydrogen gas. And looking from left to right, we see the results in E. coli, giving acid fermentation products and gas in a Durham tube. Alkalogenes faecalis does not ferment, and the tube turns red without gas. And in an uninoculated control, we just see the red color. Okay, so again, E. coli, right over here, you can see the gas. All right, and then you've got the middle tube, which is the A. faecalis. Okay, and then the uninoculated control over here on the right, no turbidity at all. Okay. And then on the right-hand side, sorbitol fermentation is a test for a pathogen called E. coli 0157:H7, which is just a designation meaning that it carries a transposon, which is a mobile piece of DNA which can move in and out of bacterial chromosomes and can confer pathogenic properties on bacteria where it integrates. Okay, the white colonies fail to ferment the sorbitol, unlike the red colonies of E. coli. Okay, so the white colonies are the pathogen. The citric acid cycle uses acetyl-CoA from the breakdown of pyruvate or other catabolites in the cell. The electrons from acetyl-CoA are used to make NADH and FADH2, while the two carbons are going to eventually be oxidized all the way to CO2, right? Remember, the end products of oxidative metabolism are water, carbon dioxide, heat, and lots of ATP, around 38 or so, okay? And so let's take a look at the Krebs cycle here.
Completely oxidized by two additional pathways. In the first, catalyzed by the pyruvate dehydrogenase complex, pyruvate is stripped of a carbon, in the form of carbon dioxide, oxidized by NAD+, forming the energy carrier NADH, and combined with a molecule called coenzyme A, forming acetyl-CoA. In the next pathway, called the TCA cycle, acetyl-CoA combines with a 4-carbon molecule called oxaloacetate to generate a 6-carbon molecule called citrate, or citric acid. Citrate is a tricarboxylate, a triple acid, and gives the tricarboxylic acid cycle its name. This pathway is also known as the citric acid cycle, or Krebs cycle. In the TCA cycle, the two remaining carbons are removed in the form of 2 carbon dioxide molecules. The energy released by the removal is captured in energy carriers, ATP, FADH2, and NADH. As a result of these reactions, the 4-carbon molecule, oxaloacetate, is reformed and can be used in another round of the TCA cycle. From these catabolic pathways, glycolysis, the oxidation of pyruvate, and the TCA cycle, a small number of ATP molecules are produced. However, NADH and FADH2 are also formed, and they will transfer their high-energy electrons to the electron transport system, or ETS. In the ETS, the energy is transformed, ultimately producing a large number of ATP molecules.
Okay, we'll talk about how NADH transfers electrons to the electron transport chain and eventually to oxygen in aerobic metabolism and aerobic respiration. We'll explain how the transfer of electrons from one oxoreductase to the next takes place and how the energy is used to establish a proton gradient. And then we'll talk about how that proton gradient can be used to make ATP. And we'll talk about how lithotrophy sends energy from reduced minerals to some species that accepts the low energy electrons. It could be oxygen, it could be some other inorganic compound or element. Okay. So electrons that are removed from substrates in glycolysis and the TCA cycle end up in NADH and FADH2 and are eventually going to be oxidized through the electron transport system, which is going to be embedded in the the inner membrane of the bacteria. Okay, the flow of electrons through this electron transport chain, which is just a series of membrane proteins that's embedded in the membrane that that using cofactors and coenzymes moves electrons from one carrier in the membrane to the next. And as those electrons move, they change their location, they lose some energy because of their change in location, right? They're changing their potential energy, and some of that energy that changes when that happens, that basically is current flowing through the chain, and that's used to pump protons into the inner membrane space. And that's eventually going to be used to synthesize the ATP, right? And that's the proton motive force that'll happen. So the space between the inner and the outer bacterial membrane is going to become acidified, and then those protons are going to flow through a very special membrane protein called ATP synthase, and as they do, that movement of positively charged particles, protons, through that membrane-bound enzyme, that's current, okay? And that current will be captured by the ATP synthase and used to create the ATP from ADP and phosphate, okay? Electron transport requires a strong oxidizing agent. It's often oxygen, okay? And respiration is going to complete the breakdown of the sugars into biomolecules. So let's look at the electron transport system.
Respiration is the overall process of catabolism from the breakdown of a substrate, such as glucose, to the reduction of a terminal electron acceptor, such as oxygen. For each glucose molecule that is catabolized from glycolysis through the TCA cycle, 10 NADH and 2 FADH2 molecules are produced. Each of these molecules contains energy that originated from glucose. Electron transport systems present in the membrane transport the electrons to oxygen, forming water, and in the process convert the energy into electrochemical gradients across the membrane. As the electrons are transferred within the ETS pumps hydrogen ions, also called protons, across the bacterial cell membrane. As a result, a gradient forms across the membrane, with a higher proton concentration outside than inside. In this way, the energy of the electrons from glucose is transferred to the energy of a proton gradient. The hydrogen ions will tend to move down their concentration gradient back into the cell, and they do so through a complex called ATP synthase. This flow of hydrogen ions through ATP synthase drives the formation of ATP from ADP and inorganic phosphate. The production of ATP using the hydrogen ion gradient from the electron transport system and the reduction of oxygen is called oxidative phosphorylation. How does the electron transport system operate? An electron transport system consists of a series of electron carriers that sequentially transfer electrons to the carrier with the next higher reduction potential, that is, to a stronger electron acceptor. Electron flow begins with an initial electron donor, such as NADH, from glucose catabolism. Ultimately, all electrons are transferred to the strongest electron acceptor, which in this example of aerobic respiration is molecular oxygen. A typical bacterial electron transport system used in respiration includes at least 3 functional components, beginning with a complex called NADH dehydrogenase. NADH dehydrogenase then transfers the 2 electrons onto the second functional component, a quinone, indicated Q. The quinone also picks up 2 hydrogen ions from the cell to balance the charge, generating quinol, QH2. The redox step from NADH through NADH dehydrogenase to quinol yields enough energy to pump 4 hydrogen ions across the membrane. Quinones and quinols diffuse within the membrane. Each quinol reduced by NADH dehydrogenase can transfer its 2 electrons onto the next ETS complex, cytochrome BO oxidoreductase. As part of the electron transfer event, cytochrome BO oxidoreductase pumps more protons. This terminal oxidoreductase now transfers the 2 electrons onto molecular oxygen. Oxygen has the strongest affinity for electrons of any other component of the electron transport system. In order to become fully reduced, molecular oxygen requires the electrons from an additional NADH molecule. In total, for every 2 NADH molecules oxidized, 16 protons are pumped across the membrane. The final 2 electrons join with oxygen and with 4 protons from the cell, producing 2 molecules of water.
So, bacterial glucose breakdown produces ATP through both substrate level phosphorylation and through the electron transport chain, pumping protons into the inner membrane space between the inner and the outer bacterial membrane. Okay, the complete oxidative breakdown of glucose to carbon dioxide and water theoretically is going to give us about 38 ATP. Under actual conditions, the amount is a little bit less. You might wonder, well, why is that? Well, because there are going to be variations in the efficiency of all these enzymes doing these jobs. Okay, enzymes that give off more heat doing this work are going to produce less ATP. And enzymes that give off less heat doing this work are going to make more ATP. Okay? And how much heat is given off by the collection of enzymes that does this series of jobs is up to the genes that code for the proteins that are the enzymes in these pathways. Okay? And there are going to be some variations in these enzymes that will make some of them more efficient or less efficient. If they're more efficient, then they're going to make more ATP. If they're less efficient, they're going to make more heat. Okay, that's one of the reasons, for instance, that you probably know people like this who can eat and eat and eat and never seem to gain any weight. Okay, one of the reasons for that can be their genetics, right? They can have extremely inefficient enzymes that do their metabolic work, and so a lot of that gets lost as body heat. Okay. There's an environmental component, of course, to it as well, right? How much you exercise and how much you eat is also going to determine your body weight, okay? The more muscle mass you have, the higher your metabolic rate is going to be, which is the rate at which you burn your body fat. And that's because muscle has more mitochondria in it because it does more work than body fat does, okay? So genetics is part of it, but environment is part of it too, okay? And the same goes for a single-celled organism, right? Okay. So we can look here, besides ATP synthase, the plasma membrane can directly drive flagellar rotation, the uptake of nutrients, and the efflux of toxins, right? So you can see the proton motive force that exists between the two membranes is power, it's energy, right? And that energy can be used to do more than just make ATP. It can be harnessed to do other kinds of work as well, okay, as a result directly of the movement of current, okay? So those are some of the examples. Some bacteria use alternate electron acceptors instead of oxygen in order to do the job. An example would be in anaerobic organisms, you could use oxidized iron. Okay, iron can exist in oxidation states of 3+ or 2+. When the iron picks up electrons, it gets reduced. Okay, so this is one way, for instance, that you could basically turn rust into iron. Okay, there are bacteria that can do a little bit of that. Okay, you can turn oxidized nitrogen into nitrogen gas, right? Pseudomonas aeruginosa is a soil bacteria and an opportunistic pathogen that colonizes the lungs of CF patients. And these are some of the pathways that go on here, right? This is an example of a Vibrio that can turn sulfates into sulfites, reducing them, okay? And again, because this is flowing through the electron transport chain, right? That current is used to establish that proton motive force, which is used to generate the ATP. And then here's another one, right? Oxidized sulfur can be reduced to um, dihydrogen sulfide. Okay, um, this in turn can be used by other bacteria to generate ATP by turning around and oxidizing the hydrogen sulfide. Okay, so that's part of the biology that happens in the hydrothermal vents. Okay, and then sulfates can be reduced to sulfites. Okay, um, or you can turn tetrathionates into tetrathionite, okay, such as seen here. Again, this is the reduced species, this is the oxidized species. So these are all anaerobes because they're not using just oxygen to be the terminal electron acceptor and could be converted into water. Okay. Lithotropy is a variation on metabolism. It's an alternative route to get electrons for the electron transport chain and generate the proton motive force. In lithotropy, an inorganic reduced molecule is the donor instead of an organic compound. So we can jack electrons from ferrous iron or ammonium or hydrogen gas, and then we can oxidize them as we saw in the previous panel, right? And those will be byproducts, okay? So they can feed the chain, all right? And then their products can be used as electron acceptors in other organisms, okay? Oxygen or an alternate gas is the final electron acceptor for the electron transport chain in these organisms, okay? It's a unique energy pathway that certain bacteria use. Example, Geobacter does anaerobic respiration. A biofilm of Geobacter can transfer electrons to a graphite electrode, and Nitrosomonas can oxidize ammonia into nitrate. Okay? We'll talk about photosynthesis now and explain its importance for human health. Okay? Photosynthesis utilizes the red and the blue wavelengths of visible light in order to generate carbohydrates from carbon dioxide and water. Okay? So it's a light-dependent pathway. There are both light and dark reactions that make up photosynthesis. The light reactions only run when the sun is up, okay? And the dark reactions run all the time. The biosynthesis is a light-independent pathway. We sometimes call those the dark reaction, where we use ATP and NADPH from the light pathways, and we generate organic molecules, okay, carbohydrates. The light's absorbed by chlorophyll and the thylakoid membranes. The absorbed energy splits water and releases oxygen, which forms oxygen gas. The light energy converts ADP to ATP, and the electrons from water reduce NADP to NADPH. ATP and NADPH are going to provide the energy for the Calvin cycle for fixing carbon dioxide and generating sugars, okay, such as fructose or glucose, okay? And this generates the biomass of the photosynthetic organism. In plants, it helps it to generate cellulose and starch and other sugars, right? In single-celled photosynthetic organisms, it's used primarily to generate simple sugars, okay? So let's take a look at photosynthesis.
Photosynthesis occurs in two stages. In the first, called the light-dependent reactions, light is absorbed by chlorophyll molecules in membrane pockets called thylakoids. The absorbed energy splits water, releasing oxygen that forms oxygen gas. The light energy also converts ADP to ATP, and the electrons from water reduce NADP+ to NADPH. These are high-energy molecules that will fuel the second stage of photosynthesis. The second stage is called the light-independent reactions, or the Calvin cycle. carbon in the form of carbon dioxide is taken from the environment and fixed into sugars and other biomass for the cell. The Calvin cycle returns the energy-depleted molecules, NADP plus and ADP, to the light-dependent reactions to become re-energized. In the light-dependent reactions, how does the absorption of light energy allow the cell to convert NADP plus and ADP into high-energy molecules? The complexes participating in the light-dependent reactions are embedded in the thylakoid membranes. The energy absorption includes two steps of light absorption, forming a Z-shaped metabolic pathway, the Z pathway of photosynthesis. The membranes contain two types of light-absorbing complexes, each containing chlorophyll molecules, called photosystem I and photosystem II. The membranes also contain electron transport systems. Light is absorbed by vast numbers of chlorophyll molecules present in the thylakoid membranes. The absorbed energy is transferred among chlorophyll molecules until it reaches specialized chlorophyll molecules in the reaction center of photosystem II. Here, an electron from chlorophyll becomes energized, which in this diagram is depicted as moving upward. This is called chlorophyll photoexcitation. Notice that the chlorophyll molecule in photosystem II is oxidized because it has lost an electron. This electron is replaced from water by a process called photolysis. Water splits to release electrons, hydrogen ions, and oxygen. An electron from water combines with the chlorophyll in photosystem II, returning it to its reduced state. In a series of redox reactions, the lost electron is transferred from chlorophyll to the electron transport system, and then from one component of the electron transport system to the next. With each transfer, the electron transfers energy to the electron transport system. Photosystem I also absorbs a photon, resulting in chlorophyll oxidation. The electron coming from photosystem II replenishes the lost electron. The final electron acceptor is a molecule of NADP+, which combines with protons and electrons to form NADPH. During its operation, the electron transport system pumps hydrogen ions across the membrane, creating a hydrogen ion gradient. The hydrogen ion gradient stores energy to make ATP. As additional rounds of the Z pathway occur, pairs of oxygen atoms form molecular oxygen. The energy carriers ATP and NADPH are now available to reduce carbon dioxide and assemble sugars through the Calvin cycle.
Now, the Calvin cycle, which are the dark pathways, the dark reactions of photosynthesis, run all the time. And what they're doing is they're utilizing the ATP and NADPH, high energy molecules, in order to make simple sugars. Okay? Examples: the Z pathway of photosynthesis shows how two photosystems with two electron transport systems obtain energy from light and make the ATP and the NADPH, and that in turn allows us to fix carbon and generate the sugars that the plant uses, the photosynthetic organism uses to serve as a source of energy and as a source of building blocks for things like the cell wall, for instance. Okay, so you can see how the transfer takes place. Right now, another interesting byproduct of photosynthesis is oxygen. Okay, and oxygen, of course, is critical for aerobic respiration. So it was the arrival of photosynthesis on the planet that changed the atmosphere from a low oxygen to a high oxygen environment. That killed off a lot of obligate anaerobic organisms, but it produced a selective pressure that allowed us to have both anaerobic and aerobic organisms on the planet for the first time. Okay, so it was one of the first examples on planet Earth of a mass extinction. Okay, in lithotropes, a form of autotrophy that is not light dependent takes place. We call this chemoautotrophy. Lithotropes are going to oxidize inorganic chemicals from the electron transport chain and make a proton motive force to generate ATP. Okay, lithotropes fix carbon using reverse TCA cycle or glycolysis or the Calvin cycle. Okay, so there are a variety of ways that we can sneak in um, the the reduced carbon. Okay. We'll talk about how intermediates of metabolism can be assembled into biomolecules, and talk about how bacteria and archaea fix nitrogen gas into ammonium and incorporate nitrogen into biomolecules. So microbes get carbon skeletons through CO2 fixation or through the breakdown of compounds that are made by other microbes. Nitrogen is obtained by nitrogen fixation or by assimilation of organic amines by doing things like consuming protein, for instance, or taking up ammonium, okay, or nitrites or nitrates. Biosynthesis spends energy from ATP and reduction energy from NADPH, right? And so these are energy consuming reactions. These are anabolic reactions. These reactions would not happen spontaneously without energy from another source, okay? And notice that they're all building large molecules from smaller pieces, all right? What do we need for cell growth as a result, right? Organic carbon for the molecules that make up the organism. That happens through carbon fixation or consuming organic carbon containing compounds. Organic forms of nitrogen, sulfur, and phosphorus are also important. The microbes can fix inorganic elements, making them available for biosynthesis. Energy transfer molecules, including ATP, NADH, and NADPH, and FADH2, okay? And biosynthesis of complex organic compounds through oxidation and reduction. Microbes fix inorganic nitrogen into a usable form, meaning a water-soluble form, right? Remember that nitrogen gas is not, it's a very stable compound. It's not available for biosynthesis. So we have to turn it into something that dissolves in water. And we do that utilizing ATP, all right, and cracking that triple bond between the two nitrogen atoms, generating two ammonias, right, some hydrogen gas as a byproduct, and then we spit out the hydrolyzed products of ATP as ADP and phosphate, right? So that's nitrogen fixation. Rhizobia are plant symbionts that convert the atmospheric nitrogen into the ammonium. They usually associate with soybeans and other legumes, beans and peas, right? And the ammonium ion is quickly incorporated into amino acids so that we don't generate toxic conditions. And the reason that inorganic nitrogen in the form of ammonia can be toxic is because it's a powerful base and can raise the pH to the point where it becomes toxic to the cell. Okay? The key tricarboxylic acid cycle intermediate, 2-oxoglucarate, or also called alpha-ketoglutarate, incorporates one molecule of ammonium at the ketone level and forms glutamate, which is a storage form for nitrogen, and it's also an amino acid, okay? And can then be used in biosynthesis of certain proteins. All right. So, in the context of an organic molecule, the nitrogen is less toxic. Okay. In the human body, most of the waste nitrogen from the breakdown of proteins and nucleic acids is in the form of a molecule called urea, where two ammonias and a carbon dioxide form this organic waste product, which ends up leaving the body in our pee, and in some cases through our sweat. Okay, so that again, the ammonia doesn't build up to a level that is toxic to our tissues and kills us. Okay, well here in the single-celled organism, we turn it into something less toxic by attaching it to an organic molecule, and we can in turn use that organic molecule for either a storage medium or to synthesize protein. Okay? So we've seen these animations. Okay, I just wanted to show you the big three that we looked at in this particular podcast. that brings us to the end of this chapter. Again, if you have any questions, get in touch with me during office hours. I'll be glad to help you out. And I will see everybody in the next podcast.